close your eyes, if they are not already closed, and let the weight of the day settle into your mattress, into your pillow, into the slow and easy rhythm of your breathing. You do not need to hold on to anything right now. There is nothing you need to remember, nothing you need to solve, nothing that cannot wait until morning, which is very far away from where you are now. Let your hands go soft. Let your shoulders drop. Let the muscles around your eyes release, and the small tensions along your jaw, and the gentle tightness that lives, without your noticing, at the base of your neck. You are here, and you are safe, and there is nothing more required of you tonight than to listen, softly, as a voice tells you a very old and very strange story. It is a story about the brain, and about the body, and about the strange and quiet ways in which the natural world can sometimes shift, silently, without warning, into something altogether different from what it was before. It is a story about a kind of illness, unlike any other illness ever recorded in the long history of medicine, an illness caused not by a bacterium, not by a virus, not by a parasite, but by something far more fundamental and far more difficult to understand. Something that lives already inside every one of us, folded quietly into the fabric of our own biology. If this voice helps you rest, consider subscribing to Plague Lullaby, but only if you are still awake. Otherwise, let this story carry you gently into the dark as we drift together, slowly, through one of the most remarkable and sorrowful chapters in the history of human medicine. It is worth beginning, as all such stories must begin, at the very beginning, which in this case is not a hospital ward, and not a laboratory, and not a city, but a hillside in rural England, sometime in the early decades of the 18th century. Perhaps around the year 1732, when shepherds in the English countryside at first began to notice something unusual and troubling in their flocks of sheep. The sheep, which had been perfectly ordinary creatures, grazing and wandering, and doing the quiet things that sheep have always done, began to behave in ways that their keepers could not explain. They would stagger as they walked, losing their footing on ground they had walked across 100 times before. They would tremble in the cold air of the English morning, shaking in ways that cold alone could not account for. They would rub themselves against fence posts and rough stone walls, with a relentless and compulsive urgency, as if something beneath their skin demanded constant friction, and it was this peculiar scratching behavior that gave the disease its name. Scrapie, from the act of scraping, the endless and futile scraping of animals who had lost something they could not name and could not recover. The shepherds of England in the 1700s did not understand what they were witnessing. They did not have the vocabulary for it because the vocabulary did not yet exist. They knew only that the affected animals declined slowly, and without exception, that the trembling worsened over weeks and months, that the coordination deteriorated, and that the sheep, in the end, died, always died, without any remedy available to their caretakers. Records from the period describe the disease with the practical, unsentimental language of agricultural communities, noting its appearance in certain bloodlines and in certain regions, suggesting to some observers that it passed perhaps through breeding, through some quality of the animal itself, though no one then could say precisely how. It would be more than 2 centuries before science would provide even a partial answer to that question, and the answer, when it finally came, would overturn nearly everything that medicine believed it understood about the nature of disease and the nature of life itself. Meanwhile, across those 2 centuries, scrapie persisted quietly in the sheep populations of Europe, a recognized and troubling, but ultimately manageable problem for agricultural communities, a disease that farmers learned to work around, without ever truly understanding. It appeared in Germany and France, in the Netherlands, and in the upland regions of Scotland, spreading slowly through flocks, resisting every attempt at explanation that the veterinary and medical sciences of those centuries could offer. Some believed it was hereditary, some believed it was contagious in the conventional sense. Some believed it arose spontaneously from poor nutrition or difficult conditions, as many diseases of the era were presumed to do, in the absence of any better framework for understanding. During this long period of uncertainty, the disease existed at the edge of scientific attention, noticed, recorded, and then set aside because there were so many other, more urgent problems competing for the resources and the energies of medicine. And yet the sheep continued to scrape, and the shepherds continued to watch, and the disease continued to move through flocks in its slow and patient way, carrying within it a secret that would not be revealed for a very long time. The secret of a misfolded protein, folded wrong in the most fundamental sense, capable of turning healthy proteins into copies of itself, spreading not through contaminated water or infected breath, but through the quiet chemistry of the body's own tissues, a mechanism that no one in the 18th or 19th century could have imagined because it required an entirely new way of thinking about what it means for a thing to be alive. During this period, across the world in a place and a circumstance utterly unrelated to the sheep pastures of England, another chapter of this long story was unfolding in the highlands of eastern Papua New Guinea, among a people called the Four who lived in the mountain valleys of the Eastern Highlands province, in a manner that had changed little over many generations. The Four were a community with deep traditions around death and the treatment of the deceased, traditions that expressed grief and respect in ways that were meaningful within their own cultural framework. And it was within these traditions that something was happening, quietly, generation after generation, that would eventually draw the attention of Western medicine in the mid-20th century and change the course of neurological science entirely. The disease that appeared among the Four people was called Kuru, a word drawn from their own language, a word that meant trembling or shivering, which described well enough the early presentation of the illness. The gentle unsteadiness that a person first noticed in themselves, or that family members first noticed in a loved one, a slight difficulty keeping balance, a subtle change in the quality of movement, a tremor that came and went at first, and then, over time, came and did not go. The illness progressed in a manner that the Four people recognized and named and understood within their own frameworks of meaning, though they could not have known and had no way of knowing. That what was happening in the brains of their sick was the slow accumulation of misfolded proteins that were consuming the tissue of the central nervous system, leaving behind a landscape of microscopic holes that would, under the lenses of mid-century microscopes, look almost exactly like a sponge, porous, hollow, devastatingly empty of the cells that had once given those structures meaning and function. The first Western physician to study Kuru in any systematic and sustained way was a young American doctor named Daniel Carlton Gajdusek, who arrived in the eastern highlands of Papua New Guinea in 1957, at a moment when the Four community was in the midst of what had become a devastating epidemic of the disease. Gajdusek was a remarkable and unconventional figure in the history of medicine, a physician with training in both pediatrics and virology, a polymath with an extraordinary capacity for fieldwork, and an unusual willingness to live fully within the communities he studied, learning their languages, participating in their daily lives, and spending years in conditions that most academic researchers of his era would have found difficult to sustain. He arrived among the four and saw immediately that what he was looking at was something unlike anything described in the medical literature, a neurological illness confined almost exclusively to one community, appearing more commonly in women and children than in men, progressing in a predictable sequence of neurological deterioration, and ending always in death, usually within 1 to 2 years of the first visible symptoms. He began sending detailed letters and observations back to scientific colleagues in the United States, letters that would eventually form part of the archival record, now held at the United States National Library of Medicine, and he performed careful autopsies on those who died, documenting the appearance of the brain tissue with the precision and thoroughness that would eventually support one of the most significant scientific conclusions of the 20th century. At the time of his arrival, Gajdusek estimated that Kuru had claimed several hundred lives in recent years, and that the disease continued to spread through the four communities with a reach that suggested something transmitted, something passed from person to person, though the precise mechanism of transmission was, in 1957, entirely unclear. It is worth pausing here, as the night settles further around you, and the sound of breathing grows slower and more even, to consider what Gajdusek and his colleagues were confronting in those early years of investigation, because the disease they were attempting to understand did not behave the way diseases were supposed to behave. It did not respond to antibiotics. It produced no fever, no inflammation of the kind associated with bacterial or viral infection. The body of a Kuru patient, examined during the illness, showed no signs of fighting anything in the conventional sense. No elevated white cell count, no antibodies mobilized against an invader, no immune response of the kind that medicine had come to associate with infectious disease over the preceding century. And yet the illness was clearly progressive and clearly fatal and clearly connected to something that passed, somehow, between members of the same community. The incubation period was another mystery, an extraordinary mystery when its true dimensions were finally grasped. Because Kuru could remain entirely invisible for years, for decades, for periods stretching in some documented cases to 40 years between presumed exposure and the first appearance of symptoms, a duration that strained the imagination of physicians trained in the rapid and visible dynamics of bacterial and viral disease. The clue that eventually broke the mystery open came not from the laboratory, at least not initially, but from the patient and careful ethnographic observation that Gajdusek and his colleague Shirley Lindenbaum brought to their study of four culture, the observation that the transmission of Kuru was linked, in a pattern that could not be coincidental, to the practice of consuming the bodies of deceased relatives during funerary rites, a practice through which the four community expressed grief and honor, and the wish to keep something of the departed close within the living, and which had, without anyone's awareness, been carrying the disease from one generation to the next, across many decades of the 20th century. Gajdusek's work took a decisive turn in the early 1960s when, drawing on a comparison suggested by a veterinary scientist named William Hadlow, who noticed a striking resemblance between the brain pathology of Kuru and the long-known brain pathology of scrapie in sheep, Gajdusek and his colleague Clarence Gibbs began a series of transmission experiments using chimpanzees. The logic of the experiment was simple, though its execution required patience across years. If Kuru was truly transmitted, as the ethnographic evidence suggested, it ought to be possible to transmit it to another animal under controlled conditions. Brain tissue from Kuru patients was inoculated into chimpanzees, and the chimpanzees were observed, and months passed, and more months passed. And then, somewhere between 14 and 22 months after inoculation, the chimpanzees began to show the trembling and the unsteadiness and the deteriorating coordination that the four people knew so well under the name Kuru. The experiment was a landmark, an absolute landmark in the history of medicine, because it established beyond reasonable doubt that Kuru was a transmissible disease caused by an agent that was present in the brain tissue of affected individuals, an agent that could move from one organism to another and cause the same devastating neurological deterioration in the new host. For this work, Gaj-Dusek was awarded the Nobel Prize in Physiology or Medicine in 1976, shared with Baruch S. Blumberg, cited by the Nobel Foundation for discoveries concerning new mechanisms for the origin and dissemination of infectious diseases. The citation was apt in its language, because what Gaj-Dusek had found was, in the most precise sense, a new mechanism, one that medicine had never previously encountered, and one that would not be fully understood until a researcher in San Francisco, named Stanley, Prusiner, turned his attention to the question a few years later, and proposed an answer so radical that the scientific community initially received it with considerable skepticism. The year 1920 is important in this story, though its significance was not recognized at the time, because it was in that year that a German physician named Hans Gerhard Creutzfeldt, working in Berlin, published a detailed case report describing a patient who had suffered from an unusual and fatal combination of neurological symptoms: dementia, involuntary movements, and a progressive deterioration of mental and physical function that ended in death, and that, on examination of the brain after death, revealed a pattern of tissue loss unlike anything previously described in the neurological literature. Within 3 years, another German physician, Alfons Maria Jakob of Hamburg, who had trained under the great neuropathologists Franz Nissl and Alois Alzheimer, described 5 additional cases with similar features, and argued convincingly that these cases represented a distinct and recognizable disease entity, a conclusion that the medical community accepted and that led to the disease being named in honor of both physicians as Creutzfeldt-Jakob disease, the name by which it has been known ever since. At the time of these original descriptions, Creutzfeldt and Jakob had no way of knowing that they were documenting what would eventually be understood as a prion disease, the most common prion disease in human beings, a disease whose mechanisms would remain mysterious for more than 60 years after their careful observations were first published in the medical journals of Weimar-era Germany. The disease they described was a sporadic condition, meaning that it appeared to arise without obvious cause in patients who had no apparent exposure to any infectious agent and no family history that suggested a hereditary pattern. And this sporadic quality would prove to be one of the most puzzling features of CJD across the decades of investigation that followed, because it meant that the body could, under certain circumstances that remained deeply unclear, generate the disease from within itself, without any external trigger. During the decades between the 1920s and the 1970s, Creutzfeldt-Jakob disease occupied a position in neurology that might be described as peripheral but persistent, a condition known to specialists, documented in the medical literature, discussed at conferences, and yet never quite at the center of mainstream scientific attention, because its rarity made it difficult to study systematically and because its causation remained entirely obscure. The disease appeared to strike approximately 1 person per million per year in populations around the world, which meant that in a city of 1 million people, one might expect to see perhaps 1 case of CJD in any given year, a rate low enough to make large-scale epidemiological study challenging but not so low as to render the disease invisible to the neurologists who occasionally encountered it in their clinics and hospitals. The patients presented in ways that were, by the time their illness was recognized, deeply sorrowful to witness. A progressive loss of memory and cognitive function, accompanied by distinctive involuntary movements called myoclonus, a jerking of the muscles that occurred spontaneously and could not be controlled, together with a rapid deterioration that distinguished CJD from the more familiar dementias of old age, and that typically brought the illness to its fatal conclusion within months rather than years. The brain, when examined after death, showed the sponge-like appearance under the microscope that had given this entire family of diseases the name by which scientists would eventually call them transmissible spongiform encephalopathies, a phrase as long and technical and quietly precise as the diseases themselves. It is worth noting, as you rest there in the comfortable warmth of wherever you have chosen to spend this night, that the story of prion diseases is in many ways a story about what medicine does not know and cannot yet see, about the gaps that exist in the best and most rigorous scientific understanding of any given era, and about how those gaps are gradually, patiently, imperfectly filled by the work of curious and persistent human minds over the long decades of research. In the 1920s, Creutzfeldt and Jacob did not know about prions because the concept did not exist. In the 1950s, the 4 people did not know about prions because no one on Earth knew about prions. In the 1960s, Gajdusek knew that something transmissible existed, something that could pass from one brain to another and cause the same devastation in each. But he could not identify what that something was, and he named it, in the language of the time, a slow virus, a term that was understandable given the knowledge available, and that was wrong in almost every important particular, as the following decade of research would eventually reveal. The something was not a virus. It was not a bacterium. It was not any of the things that biology had thus far categorized as capable of causing infectious disease. It was something new, something that the vocabulary of biology did not yet have a precise word for, something that a neurologist in San Francisco would name in 1982, with the word syllables of a longer phrase, from the syllables of a longer physis, and that word, once coined, would gradually change the way scientists thought about the most fundamental questions of life and disease, and the boundaries between the living and the non-living. That word was prion, and the man who coined it was Stanley Prusiner, and his story, which is the next great chapter in this long and quiet history, begins in a hospital in San Francisco in the early 1970s, when Prusiner encountered his first patient with Creutzfeldt-Jakob disease, and found that the experience of watching that patient decline would not leave him, would stay with him through years of difficult and controversial research, would become the organizing principle of a scientific career that would eventually be recognized. In 1997, with the most prestigious award in the sciences, Stanley Benjamin Prusiner was born on the 28th of May, 1942, in the city of Des Moines, in the state of Iowa, and he received his undergraduate education at the University of Pennsylvania, where he studied chemistry before going on to complete his medical degree at the same institution in 1968. He came to the University of California San Francisco, for his residency in neurology, and it was there, while still a resident, that he encountered a patient suffering from the devastating neurological decline of Creutzfeldt-Jakob disease, a woman whose rapid deterioration and untreatable suffering made a deep impression on the young physician and led him, when he later established his own research laboratory at the same university, to turn his scientific attention toward the question of what caused this strange and fatal illness. The scientific landscape of the early 1970s offered a clear, if not entirely satisfying, answer to that question, which was the slow virus hypothesis that Gaj-Dussek's transmission experiments had seemed to confirm. The idea that CJD and Kuru and Scrapie were caused by unusual viruses that replicated very slowly in neural tissue and produced their effects only after long incubation periods, and that the total absence of any immune response was simply a feature of how these particular viruses operated. Prusiner was not satisfied with this explanation. He began a series of systematic experiments aimed at characterizing the infectious agent more precisely, attempting to purify it, to concentrate it, to identify its constituent parts, and what he found over the course of years of painstaking laboratory work was deeply troubling in its implications, because every procedure that should have destroyed a virus left the infectious agent entirely intact. Radiation, which destroys nucleic acids, and with them the genetic material that viruses require for replication, had no significant effect on the infectivity of the Scrapie agent. Ultraviolet light, which damages DNA and RNA, did not eliminate the agent's capacity to cause disease. Chemical treatments designed to denature or destroy nucleic acids likewise failed to abolish infectivity, while treatments designed to destroy proteins did reduce infectivity substantially, an observation that pointed in a direction that seemed, when Prusiner first began articulating it, almost impossible. By 1982, Prusiner had accumulated enough experimental evidence to publish a paper in the journal Science, in which he proposed what he called the Prion hypothesis, the idea that the infectious agent responsible for Scrapie, and by extension for Kuru and Creutzfeldt-Jakob disease, was a protein, only a protein without any accompanying nucleic acid, a protein that could replicate by inducing normal copies of itself in the brain to change their shape, to fold incorrectly, to become what the disease had made of the original protein, and to go on, in turn, making more copies of that wrong shape in an ever-expanding cascade of molecular misfolding. The word he chose for this agent was prion, constructed from the first letters of the phrase proteinaceous infectious particle, with the syllables rearranged to produce a word that was pronounceable and distinct. And that word, once released into the scientific literature, became the center of one of the most vigorous and occasionally acrimonious debates in the history of late 20th century science. The opposition to Prusiner's prion hypothesis was substantial, deeply felt, and scientifically grounded in the understanding of biology that prevailed at the time. The central dogma of molecular biology, as it had been understood since Francis Crick articulated it in 1958, held that biological information flowed in one direction, from DNA to RNA to protein, and that proteins could not copy themselves, could not transmit structural information from one molecule to another without genetic intermediaries, could not, in any meaningful sense, be infectious on their own. The idea that a protein could be an infectious agent, that it could propagate through a host organism without any genetic material to guide its reproduction, struck most molecular biologists as a violation of principles so fundamental that Prusiner's hypothesis seemed to demand not just new evidence, but a wholesale revision of the foundations of the life sciences. Critics argued that Prusiner's preparations, however carefully made, must contain small amounts of nucleic acid, too minimal, to detect with the techniques then available, and that this hidden nucleic acid was the true infectious agent, the slow virus that Gajusek's work had seemed to confirm, and that all prior understanding of infectious disease required. The debate continued through the 1980s and into the 1990s. And it was not merely academic, because the disease whose mechanism was at stake was about to become vastly more visible and vastly more consequential than anyone in the scientific community had previously imagined, as a result of events unfolding simultaneously in the green and rolling countryside of England. The year 1986 was when the first confirmed cases of bovine spongiform encephalopathy appeared in the United Kingdom, identified in cattle at farms in the southern counties of England, in animals that had begun to display the same kind of behavioral and neurological changes that the farmers of the 18th century had once described in their sheep. The unsteadiness, the difficulty maintaining balance, the unusual aggression or anxiety in animals that had previously been calm, the gradual and worsening deterioration of coordination and cognitive function that gave the disease the informal name by which it would become known around the world, mad cow disease, as the newspapers and the public would call it, though physicians and scientists preferred the full clinical designation of bovine spongiform encephalopathy, or BSE, was not at first recognized for what it was. The veterinary and agricultural authorities who received the first reports approached the illness as they might approach any novel disease in livestock, gathering data, consulting specialists, attempting to establish patterns of spread and exposure, and it was through this process that a connection was gradually drawn to the feeding practices that had become standard across the British beef industry in the preceding decades. Cattle in the United Kingdom, like cattle in many industrialized agricultural systems, had been fed supplements containing meat and bone meal, a rendered product made from the processed remains of other animals, including cattle and sheep, that was added to feed as a source of protein. And it was through this feed, investigators would eventually conclude that the disease had been introduced and amplified through the national herd as infected material was processed and rendered and fed back to healthy animals, who then developed the disease themselves and contributed their own infectious tissue to the next cycle of rendering and feeding. The progression of the British BSE epidemic across the late 1980s and early 1990s was a slow accumulation of scale and consequence, each year bringing more cases than the year before, each month revealing the extent of a crisis that the agricultural and governmental authorities were, for a considerable period, reluctant to characterize in terms that might alarm either the public or the markets, upon which so many livelihoods depended. By 1988, records showed that 421 cattle in Britain had received a confirmed diagnosis of BSE, and by 1993, that number had risen to 120,000 confirmed cases, with cases at their peak reaching nearly 1,000 new diagnoses per week, a rate of spread through the national herd that no longer permitted any comfortable reassurance about the limits or the trajectory of the epidemic. The British government during this period maintained publicly, through its official advisors and spokespersons, that the disease presented no threat to human health, that cattle would prove to be what one early government committee described as a dead-end host, meaning that the infectious agent would not pass from cattle to human beings, even through the consumption of beef products. And this reassurance was repeated through the press and through parliamentary statements, with a confidence that, in retrospect, was not supported by the evidence available at the time, and that would, within a few years, be revealed as deeply and consequentially wrong. Meanwhile, in the background of these public statements, the government had by 1989 banned human consumption of certain high-risk bovine tissues, including brain and spinal cord, a measure that acknowledged implicitly the theoretical possibility of cross-species transmission, even as official statements denied any practical risk. And this contradiction between policy and public communication would become one of the most studied and debated episodes in the history of public health governance. During this same period, across the ocean, Stanley Prusiner's research program continued to accumulate the evidence that would eventually establish his prion hypothesis on firm experimental ground, though the path to acceptance was not straight, and the scientific community remained divided on the fundamental question of whether a protein alone could truly be infectious, in the sense that the hypothesis required. Prusiner's team at the University of California, San Francisco, worked through the 1980s to isolate and characterize the prion protein, which they designated PRP, and to understand the relationship between its normal form, present in the brains of healthy animals, and the misfolded form found in the brains of diseased animals, which they designated PRP scrapie or PRPSE. The normal prion protein, PRPC, was found throughout the brain and on the surface of many types of cells, and its precise biological function remained unclear, a mystery that itself attracted scientific interest because it was unusual, though not unprecedented, to find a protein so abundant and so widely expressed, whose purpose the research community could not readily identify. What Prusiner's work established, with increasing clarity through the decade, was that in diseased animals, the protein had undergone a change, not in its chemical composition, but in its three-dimensional folding, in the shape it assumed in space, and that this differently shaped form was extraordinarily stable, extraordinarily resistant to the various forces that normally break down proteins in biological systems, and extraordinarily capable of inducing the same conformational change in normal PRP molecules it came into contact with, a property that amounted, in its functional effect, to a form of self-replication without genes, the very thing that conventional molecular biology said could not happen. The recognition that PRP-NSC was not a foreign invader, but a corrupted version of the body's own protein, added a dimension of quiet tragedy to the story of these diseases, because it meant that the destruction was, in some profound sense, self-inflicted, carried out by the body against itself, by the body's own proteins turned against the tissue that had made them. In the spring of 1996, on the 20th of March, the British Secretary of State for Health stood before the House of Commons and made an announcement that changed, in a single moment, the public understanding of what the BSE epidemic had meant and would continue to mean for the population of the United Kingdom. The announcement was that a new variant of Creutzfeldt-Jakob disease had been identified, a form of the illness distinct from the classic sporadic CJD described by Creutzfeldt and Jakob in the 1920s, appearing in patients who were substantially younger than the typical CJD patient, presenting with psychiatric symptoms, depression, anxiety, behavioral changes, before the classical neurological signs appeared, and showing, in the brain tissue of those who had died, a distinctive pattern of prion protein accumulation that was similar to, and most likely derived from, the pattern seen in BSE-infected cattle. This new variant, which came to be designated VCJD, or variant Creutzfeldt-Jakob disease, represented the realization of the theoretical possibility that British authorities had for years publicly dismissed. The possibility that the Prion, disease that had devastated the national cattle herd, had crossed the species barrier and established itself in human beings, most probably through the consumption of beef products containing tissue from BSE-infected animals, consumed during the years before. The most protective restrictions had been put in place. The announcement was received with a level of public anxiety that was, given the scale of what it implied, entirely understandable, and it set in motion a period of intense scientific investigation, policy revision, and public reckoning that would occupy governments and research institutions across Europe and beyond for years to come, carrying with it a somber lesson about the relationship between agricultural practice, scientific knowledge, and the obligations of those in authority toward the populations they serve. The immediate impact of the 1996 announcement was felt across the beef industry of Europe with a swift and devastating force. As consumer confidence collapsed in a matter of days and governments across the continent suspended imports of British beef and began reviewing their own agricultural practices and their own histories of potential BSE exposure. In the United Kingdom itself, the response included the implementation of a mass culling program for cattle deemed to be at risk, a program that ultimately resulted in the slaughter of more than 4 million animals across the late 1990s, at enormous cost to the agricultural sector and at considerable emotional difficulty for the farming communities that had raised those animals and depended on them for their livelihoods. The human cases of variant CJD that had prompted the announcement continued to accumulate through the late 1990s and into the first years of the 21st century, with the total number of definite or probable cases in the United Kingdom reaching 129 by the end of 2002, according to records maintained by the Department of Health, with additional cases reported in France, Ireland, and a small number of other countries where British beef or beef products had been consumed. Each of those 129 cases represented a human life, a family, a circle of grief that extended through communities and across years of caring for people with an illness that medicine could observe but could not treat, could document but could not stop, could understand in increasing molecular detail, but could offer no remedy for, because by the time a diagnosis of VCJD was made, the process of prion accumulation had been underway in the brain for years or decades, and the tools for reversing that process did not yet exist. The science of what was happening was becoming clearer. The human cost of that science's earlier incompleteness was, in those years, arriving in the form of young people losing their minds and their lives in a manner that their families had never anticipated, and that their physicians could only witness with deep and abiding sorrow. Meanwhile, back in the mountains of Papua New Guinea, the story of Kuru was approaching its own slow conclusion, as the Four community, guided by the Papua New Guinea government and by health authorities who had worked with Gajdusek and his successors, to communicate the connection between funerary practices and the spread of the disease, gradually abandoned the ritual consumption that had sustained the epidemic across the 20th century. The decline of Kuru was not immediate, because the incubation period of the disease was so extraordinarily long that cases continued to appear for decades after the transmission pathway had been closed, people falling ill in the 1990s and even into the first years of the 21st century from exposures that had occurred 30 or 40 years earlier, during the height of the epidemic in the 1950s and 1960s. The last reported case of Kuru was recorded in 2005 in a person whose exposure almost certainly dated back to a time when the practice was still widespread, and after that year, no new cases were identified, which meant that, for the first time in what had likely been many decades, the disease was genuinely absent from the Fore community rather than merely dormant. The end of Kuru was not celebrated with any particular ceremony, partly because it could only be confirmed in retrospect, after sufficient years had passed to be sure that no more cases were waiting in the long silence of incubation, and partly because the disease had been too present, too woven into the fabric of Fore community life and Fore community grief, to allow its passing to feel entirely like relief, without also feeling, in some quieter way, like the closing of a very long and very sorrowful chapter in the life of a people who had suffered more than most from something they had not chosen and could not have prevented. The story of Kuru and the story of Scrapie and the story of CJD all converged. In the 1990s, around the figure of Stanley Prusiner and the Prion hypothesis that he had proposed in 1982, and defended through more than a decade of scientific controversy, and that convergence culminated on the 6th of October 1997, when the Karolinska Institute in Stockholm announced that Stanley B. Prusiner had been awarded the Nobel Prize in Physiology or Medicines for his discovery of prions, described by the committee as a new biological principle of infection. The award was not without its critics, because the question of whether purified prion protein could cause disease in a completely naive host, without any accompanying nucleic acid, had not been definitively resolved to everyone's satisfaction at the time of the prize, and some researchers felt that the Nobel Committee had moved ahead of the evidence in a way that was unusual for an institution known for its conservatism. But the weight of accumulated evidence from Prusiner's own laboratory and from many other research groups around the world who had spent the preceding 15 years testing and probing and extending the prion hypothesis was by 1997 compelling enough that the committee felt the discovery warranted recognition, and the decade that followed the prize would bring additional experimental confirmation that settled most of the remaining questions about the fundamental mechanism, if not about many of the finer details of prion biology that continue to be investigated in laboratories around the world today. The prize, like the diseases it honored the understanding of, came at the end of a very long journey. A journey that had begun, in a sense, on those English hillsides in 1732, when shepherds first noticed their sheep scraping against the stone walls of the countryside in the grey and quiet English morning, and that would continue for as long as there were questions remaining about how a protein, folded wrong, could travel through the living brain and leave, in its passage, only silence. The scientific community that received Stanley Prusiner's Nobel Prize in 1997 was not a monolithic body of opinion, and the debates that had swirled around the prion hypothesis for 15 years did not vanish entirely with the announcement from Stockholm, because science, unlike the awarding of prizes, does not resolve itself in a single day or a single ceremony, but continues instead in the quiet and persistent manner that has always characterized its best work, through the slow accumulation of evidence, through the patient revision of ideas, through the willingness of researchers to follow experimental results wherever they lead, even when those results are inconvenient or disorienting or contrary to what the field had previously believed. In the years immediately following the Nobel Prize, several research groups around the world turned their attention to what is called the protein-only hypothesis in its most rigorous form, asking whether it was possible to demonstrate, under completely controlled laboratory conditions, that purified prion protein without any contaminating nucleic acid could initiate prion disease in a healthy animal. The challenge was considerable because the techniques required to purify a protein thoroughly enough to exclude even trace amounts of nucleic acid were demanding and technically complex. And because the animal model systems available for testing infectivity required months of observation before any result could be reported. Over the following decade, several important experiments were conducted that moved the evidence substantially in the direction of Prusiner's hypothesis, most notably a series of studies in which recombinant prion protein produced in bacterial cells, which had never been exposed to any mammalian tissue, and therefore could not contain any contaminating mammalian nucleic acid, was shown under certain conditions to induce prion disease when introduced into susceptible mice, establishing with a degree of rigor that satisfied most remaining skeptics that protein misfolding alone was sufficient to initiate the cascade of molecular corruption that characterizes these diseases. The bacterial experiments were technically intricate, and their interpretation required care because the efficiency of infection with recombinant protein was lower than with naturally derived prion preparations, suggesting that the full story of prion propagation was not yet complete, that there were aspects of the process that the pure protein alone did not capture as effectively as the native material, and that the biology of prion diseases, however much better understood than it had been in 1982, retained dimensions of complexity that would continue to require investigation. During this period, the prion protein gene, designated PRNP in the human genome, was mapped and sequenced, and researchers discovered that mutations in this gene were associated with several of the hereditary forms of prion disease, the familial varieties of CJD, as well as two rarer conditions known as Gerstmann-Straussler-Scheinker syndrome and fatal familial insomnia, the last of which would become, in the years that followed, one of the most poignant and widely discussed of all prion diseases, partly because of its name, which carried within it a kind of melancholy poetry, and partly because of a remarkable Italian family through whose history the disease had moved for generations, in ways that had only gradually come to the attention of the medical community. The PRNP gene, in its normal form, codes for the prion protein found on the surface of neurons and other cells throughout the body, and the mutations associated with hereditary prion diseases alter the protein in ways that make it more prone to spontaneous misfolding, more likely to convert to the disease-associated conformation without any external trigger, so that in the individuals who carry these mutations, the disease arises not from exposure to infectious prion material, but from within the body's own biochemistry, from the misfortune of an inherited sequence that tilts the protein toward the wrong shape. The identification of PRNP mutations was a significant contribution to the understanding of the full spectrum of prion disease, because it explained the apparent paradox of a condition that could be simultaneously infectious, hereditary, and spontaneous. All three modes of occurrence reflecting the same underlying molecular event, the conversion of normal prion protein to its misfolded form, occurring through different initiating circumstances, but converging on the same devastating biological process. The mapping of PRNP also opened new possibilities for research. because it provided molecular targets for drug development and for genetic counseling, and because it allowed researchers to create transgenic mouse models carrying specific mutations which could be studied in the laboratory with far greater control and reproducibility than had been possible with the earlier chimpanzee experiments that Gudge Dusek and Gibbs had relied upon in the 1960s. The transgenic mouse models developed through the 1990s and into the first decade of the 21st century became one of the most important research tools in the field, allowing scientists to explore not only the mechanism of prion disease, but also the species barriers that limited or facilitated the transmission of prion diseases from one species to another, a question that had become urgently relevant in the wake of the BSE epidemic and the emergence of variant CJD. The story of the prion protein gene and its mutations was, in a sense, the molecular biography of a disease family, a family whose members shared common features and common mechanisms, while displaying, across their full range, a diversity of presentation and trajectory that reflected the extraordinary sensitivity of biological systems to small changes in the chemistry of their constituent parts. The concept of the species barrier in prion biology is one that deserves careful consideration because it is central to understanding why the emergence of variant CJD from BSE represented such a significant and troubling development, and because it illuminates something important about the way in which misfolded proteins interact with the molecular architecture of different organisms. The species barrier, in the context of prion diseases, refers to the observation that prion diseases do not generally transmit with equal efficiency between all pairs of species. That a prion preparation from a sheep with scrapie, for example, will cause disease more readily in another sheep than in a mouse, and more readily in a mouse than in a human being, and that this differential susceptibility reflects, in molecular terms, differences in the sequence and structure of the prion protein between different species. When a misfolded prion protein from one species encounters a normal prion protein from a different species, the efficiency with which the misfolded protein converts. The normal protein depends on how similar the two proteins are in their three-dimensional structure and in their amino acid sequences, because a closer structural match allows the conversion to occur more readily and more completely. The species barrier is not an absolute barrier, as the emergence of variant CJD from BSE demonstrated with painful clarity, but rather a probabilistic one, a difference in efficiency rather than a difference in kind, and the conditions under which the barrier can be overcome. The dose of infectious material, the route of exposure, the genetic constitution of the exposed individual have been and continue to be the subject of intense research in prion biology laboratories around the world. In the case of BSE and variant CJD, the barrier between cattle and humans was crossed, in the view of the scientific community, through the consumption of beef products containing central nervous system tissue from BSE-infected animals during the years before adequate restrictions were in place, with an infectious dose sufficient, in some individuals, to initiate the process of prion, misfolding in human brain tissue, that would proceed silently for years before manifesting as the behavioral and neurological changes that characterized the disease. The role of the individual's PRMP genotype in determining susceptibility to variant CJD became an important area of research because scientists observed that all of the variant CJD cases confirmed in the United Kingdom up to the mid-2000s were homozygous for methionine at codon 129 of the PREMP gene, meaning that both copies of their prion protein gene coded for the amino acid methionine at a particular position in the protein sequence, a genotype present in approximately 40% of the European population. The concentration of variant CJD cases in this genotypic group suggested that methionine homozygosity at codon 129 represented a significant risk factor for susceptibility to BSE-derived prion infection, while individuals with other genotypes at this position, those carrying valin at codon 129 on one or both chromosomes, might be either resistant to infection or subject to a longer incubation period, a finding with sobering implications, because it raised the possibility that a larger cohort of exposed individuals carrying other genotypes might develop the disease later, after incubation periods longer than those observed in the initial cases. The concern about a possible future wave of variant CJD cases in individuals with valine at codon 129 occupied public health researchers and epidemiologists through the 2000s and into the 2010s. And while the feared second wave has not materialized at the scale that some models suggested was possible, a small number of variant CJD cases were eventually identified in individuals with other PREMP genotypes, confirming that the restriction to methionine homozygosity was a matter of relative susceptibility rather than absolute protection. The species barrier story and the codon 129 story together illustrate the remarkable precision with which molecular biology in the last decades of the 20th century and the first decades of the 21st was able to descend from the clinical and epidemiological observations of disease outbreaks to the level of individual amino acids on individual proteins, finding in the chemistry of a single molecular position an explanation for patterns of disease distribution that would have seemed to a physician of the 1920s or even the 1960s entirely beyond the reach of medical understanding. Among all the prion diseases so far described in the medical literature, fatal familial insomnia holds a particular place in the imagination of those who study this family of conditions, not only because of the quality of suffering it produces, but because of the window it opens. It opens onto the role of the prion protein in the normal function of the brain. And because of the remarkable way in which the disease was tracked to a single Italian family whose history with this illness stretches back into the 18th century and who have been studied with their consent in close collaboration with researchers at the University of Bologna and other institutions since the 1980s. The disease was first formally described in the medical literature in 1986 by Elio Lugaresi and his colleagues at the University of Bologna, who reported the case of an Italian man who had developed a progressive and fatal insomnia in his 50s, an insomnia that was not the ordinary difficulty sleeping that many people experience at various points in their lives, but a total and irreversible inability to sleep, a condition in which the normal mechanisms that generate sleep were destroyed by the advancing prion pathology in specific regions of the thalamus, the deep brain structure that plays a central role in the regulation of sleep and wakefulness, and in the relay of sensory information to the cerebral cortex. The loss of sleep in fatal familial insomnia is not merely an inconvenience or a distress, though it is certainly both of those things. It is the beginning of a progressive physiological collapse, because sleep is not a passive state of rest, but an active and essential biological process during which the brain and body perform maintenance and repair functions that cannot be accomplished during wakefulness, and the deprivation of sleep leads, over time, and with cruel inevitability, to a deterioration of cognitive function, to hallucinations, to a progressive dementia, and to death, typically within 12 to 18 months of the first onset of symptoms. The thalamic specificity of fatal familial insomnia was one of the findings that made the disease scientifically fascinating, as well as deeply sorrowful, because it suggested that the particular mutation responsible, a substitution of aspartic acid for asparagine at codon 178 of the PRNP gene, in combination with methionine at codon 129 on the same chromosome, somehow directed the misfolded prion protein preferentially toward thalamic tissue, while other PRNP mutations caused their most prominent damage in other regions of the brain, a pattern of selective vulnerability that raised profound questions about how a single protein, misfolded in one way versus another, could produce such different and specific patterns of neurological devastation. The Italian family whose members had carried this mutation across many generations was studied with particular care by Lugaresi and his colleagues, who traced the disease through multiple generations of pedigree analysis and identified historical deaths in family members that, in retrospect, appeared consistent with the clinical description of fatal familial insomnia, suggesting that the mutation had been present in the family for at least 2 centuries, quietly passing from parent to child with a 50% probability at each transmission, as autosomal dominant conditions always do. The family's willingness to participate in this research, to donate tissue samples, and to allow their medical histories to be examined and published represents a form of generosity that is not always acknowledged in the scientific literature as fully as it deserves to be, because it is through the willingness of affected families to be studied and to be known that medicine learns most of what it learns about rare hereditary diseases, and the knowledge gained from studying this family and others like them has contributed substantially to the understanding of prion biology that now exists. The story of fatal familial insomnia also drew wider public attention through the work of D.T. Max, a journalist whose book on the subject, published in 2006, traced the history of the Italian family and the science of prion diseases in a way that brought these obscure conditions to the attention of readers who had never heard the word prion, and who found, in the strange quiet tragedy of a disease that takes away sleep, and then takes away everything else, a kind of dark mirror for anxieties that are far more common and far more familiar. The rarity of fatal familial insomnia, with perhaps 200 known families worldwide carrying the causative mutation, stands in contrast to the gravity of its consequences and the disproportion between its small scale in terms of case numbers and its large scale in terms of what it reveals about the biology of the brain and of sleep, is itself a kind of lesson about the way in which rare diseases, studied with care and over time, can illuminate aspects of human biology that affect far more people than carry the disease itself. The Gerstmann-Streusler-Scheinker syndrome, known by the abbreviation GSS, represents another member of the hereditary prion disease family, and its history in the medical literature is somewhat older than that of fatal familial insomnia, having been described in 1936 by the Austrian physicians Joseph Gerstmann, Ernst Streusler, and Ilya Scheinker, who reported a family in which multiple members across several generations had developed a progressive and fatal neurological disease characterized prominently by cerebellar ataxia, a disturbance of coordination and balance arising from damage to the cerebellum the region of the brain responsible for the fine control of movement, accompanied by dementia that typically appeared later in the course of the illness than the movement problems, and by a clinical course that was somewhat more prolonged than classic CJD, lasting in many cases between 2 and 10 years from the onset of symptoms to death. The 3 physicians who gave the syndrome its name were working in Vienna in the interwar period, examining a large Austrian family in which the disease had appeared in multiple generations with a regularity that clearly pointed toward a hereditary basis, and their careful clinical and neuropathological descriptions were sufficiently detailed that the case could be recognized decades later as a prion disease after the molecular tools to make that identification became available. The genetic basis of GSS was established in the early 1990s when molecular studies identified mutations in the PRMP gene. Several different mutations at different positions in the gene can cause GSS, which is one reason why the clinical presentation varies somewhat between affected families, and this discovery placed GSS firmly within the prion disease spectrum alongside CJD and fatal familial insomnia and Kuru, confirming that all of these conditions, however different in their clinical expression, and however different in the circumstances of their appearance, shared the same fundamental molecular basis in the misfolding of the prion protein. The cerebellar emphasis of GSS, like the thalamic emphasis of fatal familial insomnia, raised questions about the mechanisms by which different mutations in the same protein could produce such different topographies of damage within the brain. And these questions about selective regional vulnerability have continued to motivate research in neuropathology and molecular biology, because understanding why prion pathology concentrates in particular brain regions, in particular disease variants, is not merely an academic question, but one with potential therapeutic implications, since it suggests that the pathological cascade may have points of vulnerability that are specific to particular variants, and that might, in principle, be targeted differently. The story of GSS is in some respects a quieter story than those of Kuru or variant CJD, because it lacks the dramatic backdrop of a cultural practice or an industrial food chain, unfolding instead in the intimate and private space of affected families across generations. Each member who develops the disease confronting an illness that has already been witnessed in a parent or an aunt or a grandparent, carrying with it a particular quality of anticipated grief that is different from, and, in some ways, more burdensome than the shock of an illness that appears without warning or precedent. The genetic counseling implications of hereditary prion diseases, like GSS and fatal familial insomnia, are complex and deeply personal, because the mutations that cause these diseases are autosomal dominant, meaning that each child of an affected parent has a 50% chance of inheriting the causative mutation. And because the diseases are invariably fatal and currently untreatable, so that a positive genetic test result carries with it information of great weight and no immediate practical remedy beyond the possibility of reproductive choices that would prevent the mutation from being passed to the next generation. The families who live with these mutations, who know that they may or may not carry within their own DNA the seed of the illness that took a parent or a sibling, navigate an existential landscape that medicine created when it learned to read the genome, but had not yet learned to rewrite it. And the psychological dimension of this situation, the decision of whether to be tested and how to live with either a positive or a negative result, has been the subject of considerable attention from bioethicists, psychologists, and the affected families themselves who have shown, across the decades of their contact with researchers and clinicians, a remarkable capacity for both grief and grace. The response of governments and regulatory bodies to the BSE crisis and the emergence of variant CJD was not uniform across the countries affected, and the variation in how different national authorities managed the crisis, in how quickly they acted, in how transparently they communicated, in how thoroughly they implemented protective measures, became a subject of considerable historical and policy interest in the years that followed, as investigators and commissions sought to understand what had gone wrong and what lessons might be drawn for the governance of future food safety and public health crises. In the United Kingdom, the official inquiry led by Lord Nicholas Phillips, which published its final report in October 2000 in 16 volumes, totaling more than 4,000 pages, provided the most comprehensive account of the British BSE crisis and its handling, examining with great thoroughness the decisions made at each stage of the epidemic by veterinary authorities, government ministers, scientific advisors, and the food industry, and reaching conclusions that were in places sharply critical of the way in which the precautionary principle had been applied, or failed to be applied, in the years when the scientific uncertainty about human risk was at its highest. The Phillips report, as it came to be known, found that the government's public reassurances about the safety of British beef had not been adequately grounded in the evidence available at the time, that the precautionary measures implemented had in some cases been insufficient or insufficiently enforced, and that the culture of the government's scientific advisory apparatus had in some respects prioritized the avoidance of public alarm over the transparent communication of genuine scientific uncertainty, a judgment that had particular resonance in the context of a disease whose incubation period was so long that by the time the human cases appeared, the exposure had already occurred and the opportunity for prevention had passed. The response in France was shaped in part by the finding that variant CJD cases had appeared among French citizens who had likely been exposed through French beef products rather than through travel or consumption of British beef, and the French experience prompted its own public and political controversies about the adequacy of surveillance systems and the transparency of governmental communication about food safety risks. In the United States, where no indigenous BSE epidemic had occurred on anything like the British scale, the regulatory response was shaped by the awareness of what had happened in the United Kingdom and the desire to prevent a similar outcome, leading the Food and Drug Administration to implement in 1997 a ban on the feeding of most mammalian protein to ruminants, closing the recycling loop that had amplified BSE through the British cattle herd, and leading the Department of Agriculture to implement enhanced surveillance programs for BSE that resulted in the confirmation, over subsequent years, of a small number of BSE cases in American cattle that were managed without leading to the kind of widespread epidemic seen in Britain. Japan, which had imported British beef during the peak years of the BSE epidemic, discovered its first domestic BSE case in September 2001, a finding that prompted a major food safety crisis in the country and led to the temporary closure of all domestic beef markets, and a comprehensive review of Japanese cattle feed, regulations, and surveillance systems. The international dimension of the BSE crisis, the way in which it crossed borders through trade in animals and animal products, and through the global movement of potentially contaminated material, made it in retrospect one of the early prominent examples of a food safety risk with genuinely global characteristics, a foreshadowing of the kind of cross-border health challenges that would become increasingly central to the work of international public health institutions in the decades that followed. The landscape of prion disease research in the early years of the 21st century was shaped by two powerful and in some ways competing forces: the urgency generated by the variant CJD epidemic, which drove investment in therapeutic research and in the development of better diagnostic tools, and the fundamental scientific fascination of prion biology itself, which raised questions about protein folding and molecular conformation that had implications reaching far beyond the specific context of prion diseases. The therapeutic research was motivated by a recognition that variant CJD, once diagnosed, was invariably fatal, and that no treatment of any demonstrated efficacy was available, a situation that represented both a profound medical inadequacy and a moral imperative for the research community. Several potential therapeutic approaches were explored in laboratory and early clinical settings during this period, drawing on the growing molecular understanding of prion biology to identify points in the disease process where intervention might, in principle, be possible. One approach focused on the normal prion protein itself, reasoning that if PRP-AMC were eliminated or greatly reduced in concentration, the substrate available for conversion to the misfolded PRP-NSC would be reduced, potentially slowing or halting disease progression. An approach supported by the observation that mice genetically engineered to lack the prion protein gene entirely were found to be completely resistant to prion infection when challenged with prion preparations, a finding that suggested that the normal protein was not merely a passive victim of conversion, but an active and necessary participant in the propagation of the disease. Another approach focused on molecules that might stabilize the normal conformation of the prion protein, making it less susceptible to conversion, or that might promote the clearance of misfolded protein once formed, drawing on the cellular machinery that normally identifies and degrades misfolded proteins. Quinacrine, an antimalarial drug, and pentasan polysulfate, an anticoagulant, were among the compounds tested in cell culture and animal models, and in a small number of human patients with variant CJD, with results that were not sufficiently encouraging to establish either compound as an effective therapy, but that nonetheless contributed to the growing body of knowledge about what kinds of molecular intervention might have effects on prion biology. The challenge of drug development for prion diseases was and remains formidable, not only because of the biological difficulty of targeting a process that occurs largely within the heavily protected environment of the central nervous system, and of accessing that environment with therapeutic molecules that must cross the blood-brain barrier, but also because of the practical challenges of clinical trial design for diseases with long and variable incubation periods, small patient numbers, and no validated biomarkers that could serve as early indicators of disease activity or treatment response. The development of diagnostic tools was in some respects more tractable than the development of therapies, and the early 2000s saw significant progress in the identification of biomarkers in cerebrospinal fluid that could support the diagnosis of CJD, most notably a protein called 14-3, elevated levels of which in cerebrospinal fluid were found to be associated with prion disease and were incorporated into diagnostic criteria by the World Health Organization, alongside the characteristic EEG pattern and the clinical syndrome as a basis for the diagnosis of probable CJD in living patients. The 1433 protein was not specific to prion diseases and could be elevated in other conditions involving rapid neuronal destruction. But in the appropriate clinical context, it provided a useful additional piece of evidence for a diagnosis that, before its adoption, had often been made with less certainty during life and confirmed only at autopsy. Later, the development of highly sensitive techniques capable of detecting trace amounts of misfolded prion protein in cerebrospinal fluid and eventually in other body fluids offered the prospect of earlier and more specific diagnosis, though the clinical implementation of these techniques required careful validations across diverse patient populations, alongside the clinical and therapeutic research stimulated by the variant CJD epidemic. A parallel, and in some ways deeper set of investigations was underway into the fundamental biology of protein misfolding, investigations that placed prion diseases within a broader context of neurodegenerative conditions that share certain features with prion diseases, even if they have not traditionally been classified as infectious. The concept of protein misfolding as a disease mechanism, which prion biology had established with unusual rigor and clarity, was during this period being recognized as relevant to a much wider range of neurological conditions, including Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, and frontotemporal dementia, all of which involve the abnormal accumulation of misfolded proteins in the brain. Amyloid beta and tau in Alzheimer's, alpha-synuclein in Parkinson's, TDP43, and FUS in ALS and frontotemporal dementia, proteins that share with prion protein the tendency, under certain conditions, to adopt alternative conformations that are more prone to aggregation, and that may, in some experimental systems, spread from cell to cell in ways that bear at least a superficial resemblance to the prion propagation mechanism. The question of whether these other neurodegenerative diseases have a prion-like component, whether the spread of pathological protein aggregates through the brain in Alzheimer's or Parkinson's disease, follows the same molecular logic as the spread of PRP-ANSC in CJD, has been one of the most intensely debated and most actively investigated questions in neuroscience. During the first 2 decades of the 21st century, with significant experimental evidence on both sides, and with implications that extend to the design of potential therapies for conditions that collectively affect tens of millions of people around the world. The conceptual framework established by Prion research, the idea of a protein that can exist in multiple conformational states, one of which is capable of templating the conversion of the other, propagating pathology through a tissue in a self-sustaining cascade, has proven to be one of the most generative ideas in contemporary neuroscience, spawning research programs, funding initiatives, and theoretical frameworks that have substantially enriched the understanding of neurodegeneration beyond the narrow confines of classical prion diseases. The researchers who established this framework, Prusiner and his colleagues, and the many scientists who built upon their work in the years after the 1982 publication, could not have anticipated the full scope of the biological principle they were articulating, which has proven to be relevant not to a small family of rare diseases, but to the entire spectrum of protein aggregation disorders that represent, collectively, one of the major challenges facing medicine as human populations age. The connection between prion diseases and the broader landscape of neurodegeneration is not merely intellectual, but practical, because the tools and concepts developed to study prion propagation the transgenic mouse models, the conformational assays, the biochemical methods for characterizing protein aggregates, have found wide application in the study of Alzheimer's and Parkinson's diseases, accelerating research in those areas in ways that have benefited from the unusual scientific clarity that the prion system provides. The question of chronic wasting disease deserves attention in any complete account of prion diseases because it represents the most geographically widespread prion epidemic currently affecting any wild animal population, and because its continued spread raises questions about the long-term ecological and public health implications of prion diseases in free-ranging wildlife that have not yet been fully resolved. Chronic wasting disease, or CWD, is a prion disease of cervids, deer, elk, moose, and related species, first identified in mule deer at a wildlife research facility in Colorado in 1967, though the disease had almost certainly been present in wild deer populations before that identification was made, circulating at low levels in ways that were difficult to detect without systematic surveillance. The disease causes a progressive neurological deterioration in affected animals, weight loss, behavioral changes, loss of the normal wariness of humans that characterizes healthy deer, difficulty swallowing, excessive salivation, and a progressive wasting that gives the disease its name. And it is invariably fatal, with affected animals typically dying within months to 2 years of the appearance of symptoms. The geographic range of CWD has expanded substantially since its initial identification in Colorado, spreading across the western United States and into the Midwest, and eventually appearing in Canada, South Korea, and in 2016 in Norway, where it was detected in wild reindeer in the Nordfjella region, prompting the Norwegian government to implement an emergency culling program designed to eradicate the affected reindeer population before the disease could establish itself in the broader Scandinavian cervid population. The mechanism of CWD transmission is in some respects more complex than that of other prion diseases, because the disease can spread not only through direct contact between animals, but through environmental contamination, as infected animals shed prion material in their saliva, urine, feces, and blood, which can persist in soil and on plant surfaces for extended periods, creating reservoirs of infectious material in the environment that can infect other animals long after the original infected animal is gone. The persistence of prion infectivity in the environment is a feature of prion biology with profound implications for disease management, because conventional approaches to controlling infectious diseases, identifying and removing infected individuals, quarantining affected areas, are less effective when the infectious agent can persist for years in soil and vegetation, and because decontaminating a large area of wild habitat is not practically feasible with any available technology. The public health question raised by CWD, whether the disease can transmit to humans who consume venison from infected deer, has been the subject of active research since the 1990s, driven by the awareness that BSE had crossed the species barrier into humans, and by the fact that millions of deer are hunted and consumed in North America every year, a scale of potential exposure that would make even a low probability of transmission significant in absolute numbers. Studies in non-human primates and in transgenic mice carrying human pre-NP have produced mixed results, with some experiments suggesting that the species barrier between CWD and humans is relatively high, while others indicate that transmission is possible under at least some experimental conditions. And the epidemiological evidence, the absence as of the mid-2020s, of any confirmed human CWD cases, despite decades of venison consumption in CWD endemic areas, is reassuring but not conclusive given the long incubation periods characteristic of prion diseases. The story of prion diseases is also, in a way that is easy to overlook amid the molecular biology and the epidemiology and the policy debates, a story about animals, the sheep on the English hillsides, the cattle in the British countryside, the deer in the forests of Colorado and Wyoming, the chimpanzees in Gudge Dusek's laboratory, and the many laboratory mice who have contributed through experimental protocols carried out with as much care as the science of each era could provide to the accumulation of knowledge that now exists about these diseases. The animal dimension of this story raises questions that are not purely scientific but also ethical, questions about how humanity relates to the other species with whom we share the planet, and whose bodies and lives we have across history used in ways that have not always acknowledged the full weight of what that use entails. The cattle at the center of the BSE epidemic were not merely the victims of an industrial feeding practice, though they were certainly that. They were also the inadvertent carriers of a zoonotic risk to human health, a risk that materialized from the intersection of industrial efficiency and biological vulnerability in a way that illustrated, with particular vividness, the consequences of treating living animals primarily as components of a production system, rather than as beings whose biological integrity has implications that extend beyond their immediate commercial value. The reflective dimension of the prion disease story invites consideration of what these diseases tell us about the limits of human understanding and the risks of acting on incomplete knowledge at industrial scale. The British beef industry did not introduce meat and bone meal into cattle feed out of malice or indifference, but out of a combination of economic logic and scientific ignorance, an ignorance that was understandable in context, but that proved, when the biology of prion diseases was finally understood, to have been consequential in ways that no one at the time could have anticipated. The precautionary principle. The idea that when an action raises threats of harm, precautionary measures should be taken, even if some cause and effect relationships are not fully established scientifically, is invoked regularly in policy discussions about food safety and environmental risk. And the BSE crisis is one of its most compelling historical illustrations, a case in which the absence of a demonstrated mechanism for human harm was used to justify reassurances that, in retrospect, should instead have prompted caution and restraint until the mechanism was better understood. The long incubation periods that characterize all prion diseases added a particular and sobering dimension to this lesson because they ensured the consequences of whatever decisions were made in the 1980s would not be apparent until the late 1990s and the 2000s, by which time the exposure had already occurred on a massive scale and could not be undone, the only remaining question being how many people had been infected and how many would eventually develop disease. A question that, as the second decade of the 21st century came to its close, still could not be answered with complete confidence. The opening years of the 21st century brought with them a gradual but unmistakable shift in the emotional register of the prion disease story, a movement from the acute anxiety of the variant CJD crisis towards something more measured and more reflective as the number of new variant CJD cases in the United Kingdom declined from its peak years, and as the feared large-scale epidemic that some epidemiological models had projected failed to materialize at the scale that had been most alarming to contemplate. The decline was real, and it was welcome, though scientists were careful to note that the absence of a catastrophic epidemic did not mean that the risk had been zero, or that the decisions made during the BSE crisis had been vindicated in retrospect. It meant, rather, that the combination of the high species barrier for most PRNP genotypes, the implementation of feed bans and slaughter controls, and the particular epidemiology of the exposure had together produced an outcome that was, while deeply sorrowful for the families of those who had died, substantially less devastating than the worst projections had suggested. The total number of definite or probable variant CJD deaths in the United Kingdom reached 178 by the end of 2014, according to the National CJD Research and Surveillance Unit at the University of Edinburgh, which has maintained continuous surveillance of all forms of CJD in Britain since 1990, and whose records represent one of the most comprehensive longitudinal datasets on prion disease epidemiology in the world. The work of the surveillance unit, carried out over decades with consistent methodology and rigorous diagnostic standards, has provided not only a count of cases, but a detailed clinical and neuropathological picture of variant CJD that has been invaluable to researchers attempting to understand the full spectrum of the disease and to model its future trajectory. The cases in France, Ireland, the United States, Canada, Japan, Saudi Arabia, and other countries that were linked either to exposure in Britain or to consumption of potentially contaminated beef products added to the international picture a dimension of geographic spread that underscored the global nature of the BSE legacy and the importance of international coordination in prion disease surveillance. The slow decline of variant CJD case numbers over the 2000s and 2010s was accompanied by a continuing uncertainty about whether any additional cases were still incubating in individuals who had been exposed but had not yet developed symptoms, a question that was addressed through studies of archived surgical and autopsy tissue from the British population, studies that detected evidence of prion protein accumulation in a proportion of appendix specimens substantially higher than the number of clinical cases would suggest, leading to estimates that somewhere between 1 in 2,000 and 1 in 4,000 individuals in the United Kingdom might carry prion infection without having developed clinical disease. Numbers that were difficult to interpret in the absence of certainty about what proportion of these carriers would eventually progress to symptomatic illness. The tissue survey findings were among the most discussed and debated results in prion epidemiology during the 2000s and 2010s, and they gave rise to concerns about the potential for secondary transmission through surgical procedures, blood transfusion, and other medical interventions involving blood or tissue. Concerns that prompted blood services in the United Kingdom and elsewhere to implement precautionary measures, including the exclusion of certain donor groups from blood donation and the introduction of leukodepletion, the removal of white blood cells from donated blood as a means of reducing any potential prion burden in transfused blood products, the precautionary measures taken in response to the tissue survey findings illustrated again the distinctive challenge posed by prion diseases for public health policy. The challenge of making decisions under conditions of deep uncertainty when the worst plausible outcome is severe, but the probability of that outcome cannot be precisely estimated, and when the interventions available are imperfect and carry their own costs and complications. The science of prion strains which had been under investigation since the 1960s, when researchers first noted that different prion isolates could produce different patterns and tempos of disease in the same host species, became increasingly important in the early 21st century as a framework for understanding the diversity of prion diseases and the mechanisms of species barriers and transmission. The concept of a prion strain, which seems paradoxical in the context of an infectious agent that carries no nucleic acid and therefore no conventional genetic information, refers to the observation that different prion preparations can produce reproducible and distinct disease phenotypes, different incubation periods, different distributions of brain pathology, different patterns of PRP, NSC accumulation, that are maintained on serial passage through the same host, implying that the informations specifying these distinct phenotypes is somehow encoded in the prion preparation itself, without recourse to nucleic acid. The resolution of this apparent paradox came through the understanding that the three-dimensional conformation of the misfolded prion protein could itself vary in ways that were self-propagating, with different conformational states, sometimes described as different molecular shapes or architectures of the prion aggregate, encoding the strain information in the folding pattern of the protein rather than in any nucleic acid sequence. Different strains of prion, in this view, represent different stable conformational states of PRP-SC, each of which templates the conversion of normal PRP, say C, to a copy of itself with the same conformation, thereby propagating the strain characteristics indefinitely through successive rounds of conversion. The strain concept had important practical implications for the understanding of BSE and variant CJD, because the demonstration that the prion strain responsible for variant CJD was indistinguishable by multiple biochemical and biological criteria from the BSE strain was one of the strongest lines of evidence supporting the causal link between the two diseases, providing a molecular fingerprint that connected the human epidemic to its bovine source in a way that epidemiological data alone could not have established. The diversity of prion strains also helped explain the clinical heterogeneity observed among cases of sporadic CJD, the most common form of the disease, which appeared in several distinct subtypes that differed in their clinical presentation, their rate of progression, and their neuropathological features, differences that molecular subtyping studies in the early 2000s were able to link to different combinations of PRM genotype at codon 129 and different conformational types of PRP-NSC accumulating in the brain. The classification of sporadic CJD into distinct molecular subtypes, proposed by Pierluigi Gambetti and colleagues at Case Western Reserve University in the United States, provided a framework that improved diagnostic precision and that had implications for understanding the biology of sporadic prion disease initiation, though the fundamental question of why sporadic CJD arises, what triggers the initial misfolding event in the absence of inherited mutation or known infectious exposure, remains incompletely answered and continues to attract research attention. The most widely accepted hypothesis is that sporadic CJD arises from a spontaneous stochastic misfolding event in a single prion protein molecule, an event so rare that it occurs only once per million people per year on average, but that, once it occurs, initiates the self-propagating cascade of conversion that characterizes all prion diseases. A hypothesis consistent with the observation that sporadic CJD shows no geographic clustering, no known risk factors other than age, and no familial pattern, appearing instead with a random distribution across populations that is characteristic of a process driven by rare molecular accidents rather than by identifiable exposures or inherited susceptibilities. The molecular biology of prion propagation, by the second decade of the 21st century, had been illuminated to a degree that Prusiner himself, when he proposed the prion hypothesis in 1982, could not have fully anticipated, as structural biology techniques, including cryo-electron microscopy and solid-state nuclear magnetic resonance spectroscopy, were applied to prion aggregates and began to reveal, at atomic resolution, the precise three-dimensional architecture of the misfolded protein fibrils that accumulate in prion-deceased brain tissue. The structures revealed by these techniques were remarkable in their organization, not random tangles of misfolded protein, as earlier models had sometimes implied, but ordered fibrils with a precise and repeating internal geometry in which individual protein molecules were stacked in layers, held together by networks of hydrogen bonds and other non-covalent interactions, to form elongated structures of extraordinary stability, structures that could resist temperatures, chemical agents, and enzymatic degradation that would destroy virtually any other biological material. The stability of prion aggregates was not merely a structural curiosity, but a practical problem with significant implications for medical sterilization and for the safety of surgical instruments, because conventional autoclaving, the use of pressurized steam at temperatures sufficient to destroy bacteria, viruses, and most other infectious agents, was found to be insufficient to eliminate prion infectivity from contaminated surfaces, a discovery that led to the development of modified decontamination protocols for surgical instruments used in procedures with potential exposure to prion-infected tissue, protocols that combined high-temperature autoclaving with the use of chemical agents capable of denaturing prion protein under the specific conditions required. The structural work on prion fibrils also revealed that different prion strains, whose biological distinctiveness had been demonstrated in transmission experiments, corresponded to different structural arrangements of the protein within the fibril, different ways in which the same polypeptide chain could be threaded and folded to produce architecturally distinct aggregates that were stable enough to maintain their structural identity through rounds of fragmentation and regrowth that constituted the molecular mechanism of prion propagation. The fragmentation step in prion propagation, the breaking of prion fibrils into smaller seeds, each of which could then serve as a template for the addition of new protein molecules, and the growth of new fibrils, was identified as a critical determinant of prion replication rate and cellular machinery involved in protein quality control and aggregate management, including molecular chaperone proteins that assist in the folding and refolding of other proteins, was found to play a role in modulating the rate and efficiency of prion propagation in ways that varied between cell types and between organisms, and that helped explain some of the species barrier effects observed in transmission experiments. The structural and mechanistic insights accumulated through these years of research constituted, in aggregate, a picture of prion biology that was richer and more precise than anything available at the time of the Nobel Prize, and that served as the foundation for new generations of therapeutic strategies aimed at interfering with specific structural features or specific steps in the propagation process. As research into prion diseases deepened, and as the immediate crisis of the variant CJD epidemic receded into historical memory, a community of researchers clinicians, patient advocates, and family members of those affected continued to meet, to collaborate, and to sustain the momentum of scientific inquiry that the crisis years had generated, working through institutions, including the European Creutzfeldt-Jakob disease surveillance network, the National Prion Disease Pathology Surveillance Center in the United States, and the MRC Prion Unit in the United Kingdom, which was established at University College London in 1999, with a mandate to conduct both fundamental research into prion biology and translational research aimed at developing diagnostic and therapeutic tools for clinical application. The MRC Prion Unit, under the successive leadership of John Collinge and his colleagues, became one of the most productive and internationally recognized centers of prion research in the world, contributing to advances in prion strain characterization, therapeutic target identification, and the development of novel animal models that more accurately recapitulated aspects of human prion disease than the earlier transgenic mouse systems had allowed. Collinge and his colleagues also made important contributions to the understanding of the normal function of the prion protein, a question that had remained puzzling since the early days of prion research, and that gained renewed urgency from the observation that complete elimination of prion protein in mice produced animals that were, at least under laboratory conditions, apparently healthy, a finding that suggested the protein was dispensable for normal life, but that left open the question of whether subtle functions might be revealed under more demanding conditions or over longer periods of observation. The question of normal prion protein function was investigated through a variety of experimental approaches over the years, with evidence accumulating for roles in copper metabolism, in the maintenance of myelin sheaths around peripheral nerves, in synaptic function, and in the response of cells to oxidative stress, none of which rose to the level of a clearly defined and universally accepted function, but all of which contributed to the picture of a protein with multiple possible biological roles that might become relevant to understanding the earliest stages of prion disease pathogenesis. The therapeutic pipeline at the MRC Prion Unit and at other research institutions included, by the 2010s, several promising compounds in various stages of preclinical and early clinical development, most notably a compound called IND24 that showed efficacy in prion-infected mice and informed the development of related compounds for potential human use and antisense oligonucleotides designed to reduce the expression of the prion protein gene in the brain, an approach that had shown remarkable efficacy in animal models, and that moved into early human clinical trials, representing the most advanced therapeutic candidate for prion diseases available by the mid-2020s. The path from promising preclinical results to approved human therapy is long and uncertain for any disease, but particularly for prion diseases, given the challenges of clinical trial design discussed earlier, and the prion research community approached the therapeutic pipeline with a combination of genuine scientific optimism and the measured caution that decades of experience with difficult-to-treat neurological diseases had taught to those who worked in the field. The societal and cultural dimensions of the prion disease story did not disappear with the passing of the acute phase of the BSE crisis, but continued to manifest in ways that reflected the deep impression the crisis had made on public consciousness, particularly in the United Kingdom and in other European countries, where the experience of uncertainty about the safety of a staple food had been most directly felt. The BSE crisis contributed to a significant and lasting shift in European public attitudes toward food safety regulation, toward the precautionary principle as a basis for regulatory decision-making, and toward the transparency obligations of governments and regulatory bodies. When communicating scientific uncertainty to the public, shifts that were institutionalized in the establishment of the European Food Safety Authority in 2002, and in the revision of European Union food law to place greater emphasis on independent scientific assessment and more rigorous risk communication. The crisis also left its mark on the cultural memory of a generation of British people who had grown up eating beef and who found themselves in the late 1990s and early 2000s uncertain about what they had been exposed to and what that exposure might eventually mean for their health. An uncertainty that was, in some respects, more disturbing than a clear diagnosis would have been, because it offered no definite answer and required individuals to live with an indefinite and unquantifiable personal risk that could not be resolved by any test or medical consultation. Literary and artistic responses to the BSE crisis and to prion diseases more broadly included works that explored the unsettling quality of a threat invisible to all the senses, a disease that could not be smelled or tasted in affected meat, that produced no immediate symptoms in exposed individuals, that existed at a scale, the molecular scale of protein conformation, entirely beyond the reach of ordinary human perception, and that challenged in a deep and unnerving way the assumption that the human body can be trusted to resist and recover from the risks embedded in the ordinary business of eating and living. The philosopher and historian of science, Ian Hacking, among others, has written about what prion diseases illustrate about the relationship between classification, biology, and the human need to have named and bounded categories for the things that threaten us, observing that the prion challenged not only scientific categories but cultural ones. The category of the living, the category of the infectious, the category of the self and the other, in ways that resonated beyond the laboratory into broader questions about identity, risk, and the limits of human control over the biological world. The prion diseases also became, in the popular imagination, a kind of emblem of the uncanny. The familiar turned strange, the body's own proteins becoming agents of its destruction, the ordinary act of eating, transformed into a pathway of transmission across the boundary between one species and another. And this emblematic quality has made them a recurring reference point in discussions of biosafety, of the risks of intensive agriculture, and of the hubris that can accompany the assumption that industrial food production systems are fully understood and fully under control. The natural world, indifferent to the categories of human medicine and the timelines of human research programs, continued during all the decades of laboratory investigation and policy debate to harbor prion diseases in its animal populations with quiet persistence, reminding the scientific community that these diseases were not merely historical curiosities or industrial accidents, but enduring features of the biological landscape with which humanity would need to maintain a permanent and attentive relationship. The ongoing spread of chronic wasting disease through North American deer and elk populations represented, in this sense, an unfinished chapter of the prion story, a chapter whose ending could not yet be written because the disease was still moving, still expanding its geographic range, still establishing itself in new regions and new animal populations, and posing questions about human risk that could not be definitively resolved on the timescales of normal scientific investigation. The Norwegian discovery of CWD in wild reindeer in 2016 was particularly significant because it represented the first detection of the disease in Europe, raising concerns about the potential for spread to the large European deer and elk populations, and prompting coordinated surveillance efforts across Scandinavian countries and eventually across the European Union, which established monitoring programs for CWD in wild cervids as a precautionary measure, even in countries where no cases had been detected. The scientific community engaged with CWD was also grappling with the biological question of how the disease maintained itself in wild populations and expanded its range, asking whether the environmental reservoir of prion infectivity, the accumulated deposition of infectious material from decades of sick and dying deer, was itself driving expansion, effectively rendering large areas of habitat persistently infectious in ways that would defeat any management strategy short of the elimination of all susceptible animals, a conclusion with profound and troubling implications for wildlife conservation and for the ecological integrity of the landscapes involved. The intersection of prion disease biology with wildlife conservation introduced perspectives and priorities that were different from those that had shaped the response to BSE and variant CJD. Because conservation biology asks questions about population viability, ecosystem function, and the preservation of wild species that do not translate directly into the frameworks of food safety regulation or public health management. And because the affected animals, deer and elk living in wild landscapes, existed in a relationship with the human communities around them that was different from and more complex than the relationship between the agricultural cattle of the BSE epidemic and the industry that managed them. The prion story in wildlife was, in the 2020s, a story still being written, with surveillance data accumulating year by year, with modeling studies attempting to project the trajectory of CWD spread under different management scenarios, and with the question of human risk remaining open and actively debated in the scientific literature, a question that demanded continued monitoring and continued research. Precisely because the potential consequences of an answer in the wrong direction would be so significant and so difficult to address after the fact. The question of what prion diseases tell us about the future of human health and about the broader challenge of managing biological risks in a world of increasing ecological connectivity and industrial complexity is one that scientists, ethicists, and policymakers have been grappling with in various ways since the height of the BSE crisis, and that has not lost its relevance in the years since. The emergence of variants CJD from BSE demonstrated with unusual clarity a causal chain running from an agricultural practice, the recycling of animal protein in cattle feed, through an epidemic in food animals, the BSE crisis, to a human disease, variant CJD, a chain that was assembled over years, that was not recognized until it was already well advanced, and that could not be interrupted until a substantial amount of human exposure had already occurred, all because the mechanism of disease was not understood at the time the relevant decisions were being made. This causal chain was unusual in its specific components, but not unusual in its general structure, which is the structure of a zoonotic spillover event, amplified by human activity, a structure that has appeared repeatedly across the history of infectious disease. And that represents one of the most important categories of emerging health risk in a world where the boundaries between human populations and animal populations are increasingly permeable. The lesson of the prion diseases for emerging disease preparedness is not primarily a lesson about prions themselves, whose specific mechanisms and risk factors are now much better understood. than they were in 1986, but a lesson about the importance of maintaining scientific humility and precautionary habits of mind in the management of any biological system whose full complexity cannot be assumed to be known, and in particular, about the dangers of allowing commercial or political interests to shape the public communication of scientific uncertainty in ways that substitute reassurance for honesty. The development of better surveillance systems for prion diseases specifically, and for emerging biological risks more generally, was one of the most concrete institutional legacies of the BSE crisis, a legacy that manifested in the enhanced monitoring networks established in Europe and North America for both animal and human prion diseases, and in the broader strengthening of international health surveillance mechanisms that occurred in the first decade of the 21st century. The scientists who have devoted their careers to understanding prion diseases, Gajdusek, Prusiner, Collinge, Gambetti, Lugarese, and many others, whose names appear less frequently in general accounts, but whose contributions were equally essential, worked in the long tradition of those who study rare and difficult conditions, not only for the immediate benefit of affected patients, though that motivation was always present, but out of the conviction that the understanding of rare diseases illuminates the biology of common ones, that the molecular mechanisms revealed by extreme and unusual conditions reflect principles that operate more quietly and less dramatically throughout all of biology. The prion story, in this sense, was never merely a story about a rare class of fatal neurological diseases. It was always also a story about how proteins fold, about how information can be encoded in three-dimensional structure rather than linear sequence, about how the body's own chemistry can become the source of its own destruction, and about the extraordinary capacity of the human mind, working slowly and collaboratively across decades and continents, to find in the most perplexing biological phenomena the beginnings of understanding. There is something almost meditative in the thought of the prion protein itself, that small and ancient molecule present on the surface of cells, throughout the nervous system of every mammal that has ever lived, folded into its normal configuration by the biochemical environment of a healthy cell, carrying out whatever quiet functions biology has assigned to it across hundreds of millions of years of evolutionary history, unaware. If a protein can be said to be unaware of anything, of the catastrophic alternative that lies latent in its own structure, the possibility of misfolding into a form that propagates itself at the expense of the tissue that made it. The normal prion protein, PRPC, is encoded by a gene present in the genome of every vertebrate species studied, which suggests that it serves some function important enough to have been conserved by natural selection across the long sweep of vertebrate evolution, and yet the disease-associated form, PRPNSC, is in the most fundamental sense a corruption of that function, a protein that has forgotten, if one may use that word, the shape in which it was supposed to exist and adopted instead, a shape that slowly and irreversibly dismantles the very tissue from which it came. The evolutionary perspective on prion diseases raises questions that are not easily answered. Why has natural selection not eliminated the misfolding susceptibility of prion protein more completely, given the devastating consequences of that susceptibility when it manifests? And the tentative answers suggest that the misfolding susceptibility is either a necessary consequence of the protein's normal functional architecture, a structural compromise inherent in the same features that make the protein useful in its normal form, or that the rate of spontaneous misfolding is sufficiently rare. in the context of most individual lifetimes, to have exerted insufficient selective pressure to drive further resistance, or some combination of both. The thought of a protein that has existed for hundreds of millions of years, largely harmless, occasionally catastrophic, present in the brains of every person who has ever lived and whoever will live, waiting in its normal form in each neuron for the rare and random event that might initiate its conversion, has a quality of quiet uncanniness that is different from the more familiar uncanniness of viruses and bacteria, which are at least clearly alive and clearly other, clearly distinguishable from the self. The prion is not other in that way. It is made of the self, it is part of the self, it is the self turned by the smallest possible molecular change in shape into something that the self cannot survive. There is, in that biological fact, something that invites a kind of philosophical reflection, a reflection on the fragility of the boundary between the helpful and the harmful in all of biology, on the ways in which the same molecules and the same mechanisms that sustain life are always, by some narrow margin of chance or chemistry, potentially the mechanisms of its dissolution. The researchers who have spent their lives studying prion diseases have not, for the most part, expressed their work in these reflective terms, because the culture of science prefers the language of mechanism and evidence to the language of contemplation. But the work itself, accumulated over decades, has produced a body of knowledge that invites exactly this kind of reflection. A reflection on what it means to be a living thing in a world where the boundary between structure and dissolution is maintained not by any force of will, but by the quietly improbable stability of correctly folded proteins doing their work in the darkness of the living brain. The current state of knowledge about prion diseases, as the third decade of the 21st century advances, represents a remarkable distance travelled from the bewilderment of the English shepherds of 1732 and the confusion of the neurologists of the 1920s and the limitations of the researchers of the 1960s, a distance measured not in miles or years, but in the accumulated understanding of a biological phenomenon that had no precedent in the history of medicine, and that required, for its full comprehension, the development of entirely new conceptual frameworks and entirely new experimental tools. The understanding that exists today encompasses the molecular structure of the misfolded prion protein, the mechanism by which it converts normal protein to its own conformation, the genetic factors that modulate susceptibility and disease expression, the epidemiology of acquired, hereditary, and sporadic prion diseases, the pathological changes that occur in the brain as prion disease progresses, the strain diversity that underlies the clinical heterogeneity of these conditions, and the public health measures appropriate to the management of prion risk in both food safety and medical contexts. And yet, alongside this substantial body of knowledge, the questions that remain open are themselves substantial. Why the normal prion protein exists and what precise functions it serves. How the extremely long and variable incubation periods of prion diseases are biologically determined. Why prion pathology concentrates in different brain regions, in different disease variants. Whether and under what conditions chronic wasting disease poses a risk to human health, and, most pressing from the perspective of those affected, whether it will ever be possible to develop a treatment that can halt or reverse prion disease in a living patient, offering something more than the careful and compassionate management of an irreversible decline. That is currently the only option available to clinicians caring for prion disease patients and their families. The therapeutic research that is ongoing in several centres around the world And that has advanced furthest in the development of gene silencing approaches designed to reduce prion protein expression in the brain, represents the most tangible current hope for a meaningful clinical intervention, though the translation from animal models to human patients involves challenges of delivery, safety, and timing that are formidable and that will require years of careful clinical investigation to resolve. The families living with hereditary prion diseases, who know that a mutation is present in their genomes, and that the disease may come for them as it came for their parents, follow this research with an attention and an investment that goes beyond what most scientific observers can fully appreciate, because for them, the question of whether a therapy will be available is not abstract, but deeply personal, measured not in academic interest, but in the possibility of living long enough to see a grandchild, or of remaining oneself long enough to say a proper goodbye. The prion disease story is, in its final dimension, a story about people, the scientists and the patients and the families and the communities, and about the slow and difficult and sometimes beautiful process by which human beings, confronted with a mystery they do not understand and a suffering they cannot immediately relieve, choose to keep looking, keep asking, keep working, in the faith that understanding, arrived at carefully and honestly, is the most durable foundation for everything that might eventually come after. The laboratories where Prion research continues today are quiet places, as most laboratories are. Rooms filled with the soft hum of centrifuges and the gentle light of microscope screens, where researchers spend long hours with materials that require patience and precision, and a willingness to work at timescales that do not conform to the urgencies of ordinary life. The mice in their cages, the cultures in their flasks, the protein preparations in their tubes, all of them carry forward the investigation that began, in one sense, in the highlands of Papua New Guinea in 1957, and in another sense on the sheep pastures of England in 1732, an investigation that has been sustained across generations of researchers by the quiet persistence that characterizes the best of scientific work. The knowledge accumulated in those laboratories has moved slowly into the clinical world, into the diagnostic criteria used by neurologists when they evaluate a patient with unexplained dementia, into the decontamination protocols used by surgical teams, when instruments may have been in contact with neural tissue, into the blood safety measures implemented by transfusion services, into the surveillance networks maintained by public health agencies in dozens of countries, changing in practical and sometimes life-saving ways the experience of people who will never know the word prion and will never need to. The story of prion diseases is not over, because the diseases themselves continue, in their various forms, to affect the small number of people who develop them each year, people who deserve and receive the best care that medicine can currently provide. And because the science continues, in its patient and cumulative way, to move toward the understanding and the interventions that future patients may benefit from, in ways that current patients cannot. And so the research continues, and the surveillance continues, and the quiet accumulation of knowledge continues in those lit rooms in San Francisco and London and Bologna and beyond, where people who care about these questions spend their working lives in the company of a protein so small it cannot be seen by any ordinary means. A protein that has taught medicine more about the nature of disease than almost anything else in the long history of its investigation. And here, in the slow drift of this late hour, as the words grow softer and the spaces between thoughts grow longer, It is enough to know that the science is in good hands, that the questions are being asked, that the people who were lost to these diseases are remembered by those who study them, and that the long arc of human curiosity, patient and persistent as it is, continues to bend, however slowly, toward understanding. The prion is still there, folded correctly in the neurons of every person alive, doing its quiet and mysterious work in the chemistry of the living brain, and the vast majority of those proteins will fold correctly for every moment of every person's life and will never cause any harm at all. The risk is real, but it is rare, and the knowledge that now surrounds it is vast. Compared to what existed even 30 years ago, and the people working on it are careful and dedicated and will keep working. You do not need to hold any of this. You do not need to remember the names or the dates or the mechanisms. You have carried the story this far, through all its chapters and all its centuries, and now you can let it go. Let it drift away like the soft sound of a voice in another room, like the distant toll of a bell in a city you have never visited, like the memory of a dream that was vivid a moment ago, and is already, very gently, beginning to fade. The prion diseases belong now to history and to science, to the researchers and the clinicians and the families who carry them forward. And you belong here, in this moment, in this room, in the slow and easy rhythm of your own breathing, which is steady, and which is yours, and which will carry you, if you let it, all the way through to morning.